All right, so DNA replication today. This will not be one lecture. This will probably be 1.5 to 2 lectures. So we're not going to get through all of this today, but I'm hoping to get to a certain point, and we'll see. We've got lots to talk about DNA replication, and it is there's a whole bunch of enzymes and proteins that we got to talk about too. So let's get into this. So you may remember last day we were talking about Watson and Crick and their discoveries and the discovery of the structure of DNA, and we were talking about kind of the basics around DNA replication. And the basics are you have a parent molecule, the hydrogen bonds in between the nitrogen spaces get separated, and then we can replicate one of the strands. And so an enzyme called DNA polymerase is going to come in and it's going to match our nucleotides with complementary nucleotides. So A pairs with T, C pairs with G, and so on. And so here it goes with that little strand there. So that's what we're going to talk about today. And so there's a few things to kind of discuss before we get into all the details. So first thing to discuss is it says here, what does it mean that the two strands of DNA are complementary and anti-parallel? Okay, so two things to consider. And this is kind of a good exam question, by the way. So let that be a hint, something I considered in some years. So what do those two things mean? Complementary. So complementary means one thing. It means that we have the matching base pairs, right? So if you remember these rules, right? A, adenine pairs with T, thymine, and they have 2 hydrogen bonds. So there's our hydrogen bonds right there, those little dotted dash lines. And our G pairs with C. So that's what complementary means. It means we have one strand is complementary to the other strand. It's not identical, it's complementary. It has that base pair matching going on, right? So what does anti-parallel mean? So that's not the best picture. I'm going to draw one for you, a picture of deoxyribose. So deoxyribose looks something like this. So it is a pentagonal structure, and it looks something like this. It's got a couple of alcohol groups on it. So here's the thing, right? We've got this 5-carbon sugar, right? And it's got, so 1, 2, 3, 4, and 5. So this is something that sometimes we do. We number carbons on a chemical structure. And the reason that we number carbons is, well, sometimes we want to talk about them. We want to say, hey, what carbon is the phosphate attached to? Or what carbon is the nitrogenous base attached to? And so we number them, and organic chemists do this all the time. They might talk about carbon number 3 on this molecule, or carbon number 7, or whatever. In this case, we have 5 carbons, so we number them 1 to 5. And so the thing is, what if we're talking about carbon number 3? And then the next question is, are we talking about carbon number 3 on the sugar, or are we talking about carbon number 3 on the nitrogen base? So to distinguish them, they give them these little ticks. These little ticks are called primes. And so now we're going to talk about not just our carbon number 3, but we call it 3' carbon. So it basically means carbon number 3. So if you take a look at this here, and I'll show you a little bit better of a diagram here. Oh, I didn't realize I had writing there. I apologize. So each of these strands has a little bit of a polarity to it. So let me just get rid of that for a second. So notice that at carbon number 5, this is where the phosphate is attached to. And over here at carbon number 3, that's where the next nucleotide is going to be attached to. So let me show you the picture there. I'll come back to that again right now. And you can see that we have this little strand. I'm looking at the strand here on the left, and this strand here. And so this is basically the chemistry of this whole thing. We say the strand runs in the 5' to 3' direction. So it's kind of like thinking about the English language, right? We read things left to right. Well, this is the way we read nucleotides, from 5' to 3'. And this is just, like I said, has to do with the chemistry. So notice we talk about this here having a 5' end and a 3' end. So notice the other strand, our complementary strand, runs in the opposite direction. So this one here is 5' to 3', and this strand here runs in the opposite direction. So 3' to 5', or you can think of it as just flipped upside down. So that's what we mean by anti-parallel, is one strand is in the opposite direction to the other strand in terms of how the chemistry is going. And like I said, when Watson and Crick were doing this, they learned that this is the best way. The base pairs, they match up this way. They don't match up if you flip them up the other way around, right? Okay, so I do have some arrows. There we go, some animations. So there's an implication to this. It turns out this 5' to 3' thing. We're going to talk about 5' to 3' again and again and again over the next few units. Like I said, think of this as just, this is the direction that DNA and RNA, it goes in. It's the proper direction. So if you take a look at this, we want to make some new DNA. Turns out that the enzymes can only work on this 3' OH. So this is kind of like the substrate of the enzyme. That's what it recognizes. So the new enzyme is going to grab one of these things here. So you're probably wondering, what is that thing there? That thing there is a DNTP. So what is a DNTP? A DNTP is a deoxynucleotide triphosphate. So it turns out there's 4 of these, right? We've got DATP, we have DCTP, DGTP, and which one am I missing? TTP. Okay, there's the 4 letters of the genetic code, A, C, G, and T. So these two things, this 3' OH and this deoxynucleotide triphosphate, those are the substrates of the enzyme. And so it turns out that you can see maybe up here in this note at the top, it says nucleotides are added to the chain in the 5' to 3' direction. So these enzymes only work in one direction. They only go 5' to 3'. So like I said, that's why we're going to be talking about this 5' to 3' stuff again and again. And the enzyme that does this is called DNA polymerase. And I'm going to make some notes about DNA polymerase in a minute. So DNA polymerase, it's making a polymer of DNA. So it's a good name for an enzyme. So there's our new nucleotide, and you can see that it goes from being a DNTP, a triphosphate. It loses 2 phosphates here. That's going to get recycled, and the new nucleotide is added to that strand of DNA. So a few more things to talk about before we get into the details. First thing to talk about is that where this replication begins. We call this a replication fork. So you can see this replication fork. We have one strand up here, and it basically gets split into 2 strands, right? And the 2 strands become 2 new strands through the addition of newly formed DNA molecules. So if you are a bacterium, you're going to have a circular chromosome, and replication starts at this place called an origin of replication. So an origin of replication, what exactly is that? It's actually a DNA sequence. And it's a DNA sequence that says to the enzymes, hey, come over here, start here. And so that's what it does. So what happens is that the DNA starts to separate at that origin of replication, and new DNA is going to be made. So you can see in this little diagram here, we've got a little bit of new DNA being made. The daughter strands are represented by the lighter blue color. And it basically goes in both directions, right? You're splitting this apart. Replication go left, replication can go right. And so we have actually 2 replication forks, and we call this a replication bubble, by the way. So it keeps on going around the entire chromosome. Eventually what's going to happen is it's going to, they're going to meet, and you're going to end up with those 2 daughter DNA molecules. Here's an electron micrograph of a replication bubble. You can see the 2 replication forks represented by the arrows there. So for bacteria, this is they have chromosomes that maybe 4 million base pairs. It's not massive. It's big, but they can do this in about 30 minutes or so, kind of depending on the size of the chromosome. And so they only need 1 origin replication. So there's a I'm going to talk about eukaryotes, but one thing I wanted to mention is that these origin replications are really important for any DNA molecule that you want to be replicated in an organism. So they're going to have these on chromosomes, and they're going to have them on plasmids. So there's an implication of this, meaning that if you are in the biotechnology industry and you want to make an artificial plasmid, all you need is an origin replication. So this is actually how we make artificial plasmids. We need that origin replication, so it's a little bit of a DNA sequence. You can add it to a circular DNA molecule and you get this final thing that we call an artificial plasmid, which is something we can replicate in E. coli, for example. And that's what was done with last week's lab. It was an artificial plasmid that was produced and replicated in E. coli. So here is a plasmid, and you can see that plasmids are something called supercoiled. So supercoiled, it means that your normal plasmid is not just a big hoop like this, but the DNA sort of winds itself and twists itself a little bit just to be a little bit more compact. And I thought I'd show you that picture. So what if you're a eukaryote? You're a eukaryote, you have massive chunks of DNA. And so as a consequence, eukaryotes have multiple origins of replication. So same idea, they make these little replication bubbles. Replication goes to the left, to the right. Eventually, the replication bubbles, they're going to meet each other. And there we go. And then you get your DNA is going to be replicated. And there's a whole bunch shown in a micrograph there. You can see those replication bubbles. And you'll end up with two daughter DNA molecules. Okay, so, oh, there's a nice, another nice micrograph showing all those replication bubbles. So I told you I wanted to talk about DNA polymerase. This is kind of the enzyme that does all the important stuff, or much of the important stuff for replication. So I have a bunch of notes that I'm going to write down regarding this enzyme, and they all have implications in terms of how this enzyme actually works. So first thing to note is something I told you already, is that this enzyme has two substrates. So it has two substrates. Maybe, can you remember what they are? One was the DNTPs. So the deoxynucleotide triphosphates. And of course, there's four of them. I'm not going to write it all out again, but A, T, and C, and G, of course. And the other substrate is the 3' end of the growing strand. So 3' end of the growing strand. Okay, so as a consequence, there's a couple of things we need to note about that, right? So DNA polymerase, I'm just going to abbreviate it as DNA pol. It cannot start replication without that 3' end. So it's going to need a start. And we call that a primer, this molecule, we call it a primer. So 3' OH primer. We'll get into a bit more detail on that in a few minutes. So like I said, it can't start out of nothing. It's got to have something to grab onto. It has nothing to grab onto, it can't get started. So sometimes this enzyme needs a little bit of help from something called a primase. That's another enzyme we'll talk about in a minute. So as a consequence of this 3' end thing, like I said, it synthesizes DNA in one direction, and that's the 5' to 3' direction. So it synthesizes the new strand in the 5' to 3' direction. Okay, so that's not it. I've told you what the substrates are. I've told you it can't start without help. And so that's kind of the beginning. I have about maybe 3 more notes on DNA polymerase. So hopefully you got all that. And I'll just review the video if you didn't get all that. But I'm going to go to the next note. So the next note is something that you should probably know already, is that the new strand is complementary to the template strand. So the new strand is made complementary to the template strand. So the template strand is the old strand, the one that's used by the enzyme to decide which nucleotide to put in. Okay, a couple other notes. I just got to find them here. Make sure I got everything here. The last couple of notes are just kind of like by the way kind of notes. So one interesting thing about DNA polymerase is that DNA polymerase, DNA pol, has proofreading activity. So that means if it makes mistakes, it can do things. Proofreading activity. So it can remove incorrect nucleotides. And the last note is this here, is that DNA polymerase requires other enzymes and proteins for help. And I think I said that already. Other enzymes, proteins, we'll say to function properly. Okay, hopefully you got all that. Looks like a little bit of a mess there. I just, writing on screen, I really apologize. Handwriting is much neater, but at least I know my handwriting is neater than Soren's. I know some of you have him for chemistry, and I tell you, I at least have comfort in knowing that my handwriting is better than at least one person out there. So, yeah, let's talk about some of the details on how DNA replication works. So as I mentioned, this is kind of similar to that diagram that I showed you before. And you can see this is just a different diagram. This is the one from the textbook. And you can see we've got that free 3' end over here. Here is the DNTP, the nucleotide that's coming in. And so the next base you can see is an A on the template strand. And so it's going to match it with the T. So there goes my T. It's going to form those hydrogen bonds. And the DNA polymerase is going to take that substrate, the DNTP. It's going to remove 2 phosphates and attach that nucleotide onto the previous one at the 3' end. So now it's a little bit longer. So what I want to do is kind of talk about this process in a bit more detail and talk about some of these accessory enzymes. And then what I'm going to do is draw you a big picture on with notes on the whiteboard. And hopefully that will all help it come together kind of at the end once we go through everything. So here is a picture showing the DNA replication bubble, right? And we're going to start off and we're going to zoom in on that box over there on the left. And so notice that the dark blue, that's the parent template strands, and the light blue are the light blue is the new DNA that's being made. So first thing I want to point out is this thing here. So like I said, I'm just looking in the box for now. And remember I told you that DNA polymerase cannot start by itself. It can't just grab onto nothing. So what we do is we have an enzyme called primase, and I will define primase. Try that again. Primase. I will define primase in a few minutes once I do my whiteboard diagram. But primase basically puts a little chunk of RNA there. So why it uses RNA, I don't really know, but it works for the cell. But RNA is like DNA, it has that extra 3' end. So it's just kind of a little bit of a starting point, just to get things going, get the wheels greased and get this thing going. And so the enzyme can attach, and then it's making its DNA, and you can see it's going here in the 5' to 3' direction. So just ignore that one for now. So this was a 5' up here, and 5' to 3' direction. And there's the new DNA. And so this replication fork is just going to keep separating, and that new DNA is going to continue to be formed until it comes to the end of, meets another replication bubble. So let's talk about what's going on in that area there, and we call this the leading strand. And I'll talk about what the lagging strand is in a few minutes. So to get things going, I mentioned the primase right there, and there's a few other things that are important. Okay, and so in fact we're going to have 6 enzymes and proteins to learn for this particular unit. So the primase gets things going, puts in this RNA primer here. We also have this thing here called the helicase. So the helicase is basically in there and it's breaking hydrogen bonds. It's separating out the 2 strands of the double helix. So you can see it's separating things out here. I kind of think of it as like a little wedge or an axe or something that's just kind of going in there and chopping out those hydrogen bonds. The DNA wants to kind of reattach to itself. And so we have these single-stranded binding proteins in there that basically hold the DNA in a single-stranded conformation until the DNA polymerase can get in there. And then the last thing we have is this one here called a topoisomerase. And sometimes it's also called a gyrase. So this is actually upstream. And so this is kind of like an untangling enzyme. So if you think about it, maybe some of the ladies out there have had their hair braided. And to unbraid your hair, you have to carefully, take it apart. If you don't, if you just try to yank at it, it's going to tangle up. It's the same idea. If you ever take a piece of string, try pulling apart the strands, and you see it'll knot up. Because the DNA is a helix, right? And so if you try to unwind it, it gets tangled. So the topoisomerase is kind of like an untangler kind of enzyme. And I'll talk about that as well in a few minutes. So like I said, I'm going to do a nice diagram here in a minute and define all these enzymes and write a little note about each of them. So here is the leading strand. So the primase has already made our RNA primer over here. And this here is DNA polymerase. There are more than one type of DNA polymerase. I'm not going to concern myself with it in this class, but you can see that this is DNA polymerase 3, and it's synthesizing DNA. So all those notes that I just told you before, it's going in the 5' to 3' direction. It's adding the new base pairs are going to be complementary, and so on, and it's just going to go. So there it goes. It's going a little bit further. It's made a bunch of attached a bunch of nucleotides together, all in the 5' to 3' direction. So let's, what is this here? This is just similar diagram, I guess. Okay, I must have found a different diagram here. I don't need to show you this one here as well. So let's talk about the lagging strand and what is going on here at the lagging strand, because it's a little bit, you got to think about what's going on. So just take a look on the left here. Just ignore all that stuff for now, okay? So our DNA, it's separating, right, in this direction. But it would have started separating here at the origin of replication. And so it separates maybe this much here. But the DNA can only be made in the 5' to 3' direction. So down here, we have this strand here is 3'. 5' is over here. So these little strands here, they can only go in one direction, 5' to 3'. So it opens up a little bit, and this one here is made first. And then it opens up a little bit more and makes a second little fragment, opens up a little bit more and makes another fragment, and it's just going to keep making these little fragments. And these little fragments are called Okazaki fragments. And so it's just because that enzyme is limited by the direction it can go in. So let's take a look at this here, and I'll show you a little bit more detail on this. And like I said, I'm going to do a diagram here in a moment. So first we have our primase. So same as before, primase makes a little primer. Our DNA polymerase is going to get in there, and it's still DNA polymerase 3, but like I said, don't worry about the 3, and it's going to form a little fragment. Eventually, at some point, it's going to bump into the next fragment, and so it actually detaches. And you can see there's another fragment being made, and so on. And so you may notice that at this point here, we have all these fragments, and unfortunately, we have a bunch of RNA here. And so it turns out that the DNA polymerase can actually remove that, and it's actually a different DNA polymerase, but it doesn't really matter. It's removing that RNA. And one problem is eventually it's going to reach right here, and there's going to be a nick. It can't finish that gap. And so there's another enzyme called DNA ligase that's going to go in there, and it's basically going to seal up the gap between the two strands. Okay, so here's our DNA replication fork. And like I said, what I'm going to do is draw something for you right now. And I'm just going to share my whiteboard, and hopefully it'll make a little bit more sense once we start drawing this out together, okay? So let's take a look at this. What I'm going to do for you is I'm going to draw some DNA, okay? And so I'm going to draw it like this. And I guess in this case here, what I'm doing is I'm drawing the other strand on the other side of the DNA. So the other side of the fork. So what I've done is I've drawn a fork here, okay? And so our DNA has that polarity. It's got the 5' to 3'. Okay, and so what I'm going to do is kind of make these little arrows on the ends of each of these, because I'm thinking of the 3' as the end, so it starts as the 5'. Okay, so there's my DNA. Okay, so what I'm going to do now is start filling in some of the details of what is needed here. So first detail is going to be my helicase. So here is my helicase, and I'm going to draw it in right here like this. So there's my helicase, and color that one nice and green. And I have a note here that I told you I was going to write a little bit about a note for these. And rather than making you bear my handwriting a bit more, I'm just going to cut and paste actually. So there's my helicase note right there. So sorry, I'm just going to shrink this a little bit. And there is my helicase note. So I guess I meant to draw the whole diagram a little bit bigger, but that'll do, I think. So there's my helicase, and you can see my note. It says helicase, it separates the two strands of DNA by breaking hydrogen bonds. Okay, so the next thing I need a new color. Let's go with purple, I guess. We have these single-stranded binding proteins, so I'm going to put them in like this. So single-stranded binding proteins. You can call them SSBP if you want. So what are these guys doing? There's my note there. So single-stranded binding proteins. I'm going to move that up there. Maybe up top is a good place to put it. Okay, single-stranded binding proteins right there. So these are, like I said, they're keeping the DNA single-stranded. So that's what the SS here stands for, single-stranded, and prevents them from becoming double-stranded again, right? So that's the first part. This whole thing gets separated. Okay, so maybe another note to put is that I'm going to put a little arrow over here like this. Okay, and this is going to be the direction of DNA replication. So direction, DNA replication. So next thing to throw in is my DNA polymerase. So I'll find a nice color for that one. Why don't I make that one, I don't know, orange or pink or something like that. There's orange. Okay, so my DNA polymerase, I'm going to stick it in right here like this. Okay, and what I need to do, I think, is just move this here up out of the way a little bit. And my new DNA, I'm going to do in black. How about? So here we go with my new DNA. So remember, it would have started over here wherever the origin replication is, and I'm just going to draw it like that. And it's kind of going in this direction here. And so that would be my 3' end there. So here are my new strand. And that's my DNA polymerase. So I got a note for you for the DNA polymerase. I know we have a massive note for our DNA polymerase, but we're just going to do the small note, which is saying that it's adding the new nucleotides. So we'll do that. There it is. There's my note. Don't know why it likes to post it right in the middle for me. There we go. So DNA polymerase, it says here, go. It adds new nucleotides at the 3' end. Okay, so this here is the leading strand. So that's the other note. So as this helicase separates out the double strand, the DNA polymerase is going to go along and it's going to add those new nucleotides. And that's kind of it. Just don't know why I can't get my cursor out of that box. So hold on a second. There we go. Okay. And that's pretty much the leading strand. Now, there's one other thing to mention with the leading strand is that we have that topoisomerase. So I will do that in a blue color over here. We'll make the topoisomerase. There we go. I'll make it a nice light blue color. So remember that that is kind of like an untangling enzyme. So let me just get my note for that. Topoisomerase. There we go. I'll put that note over here. Sorry, I keep zooming in and zooming out. It doesn't, sometimes it does that automatically for me. So topoisomerase right there. So it says here, it untangles and reduces the tension in the unwinding strand. Okay, so that's the leading strand. And I think we have just enough time to draw the lagging strand for you. So let us draw the lagging strand. So like I said, we want to think about this as it started at the origin of replication, okay? And it can only make the DNA in the 5' to 3' direction. So what I'm going to do is I'm going to draw the first strand here. So I guess our new DNA is black, so I've got to make sure I have the right color. So the very first strand would have been made way over here on this side. So 5' to 3'. So 5' to 3'. And I'm going to draw a couple of strands here, and I'm going to come back to that in a moment. Okay, so notice there's no RNA there because since that strand was made earlier, the RNA is gone already. So maybe what I need to do is draw a couple other strands here. So the next strand, sorry, I just got to change my color here. So here's the next strand here, and it's going to be made with a little piece of RNA. So there's my RNA primer, and the RNA primer has a 5' to 3' end. So that says 5' to 3'. And then we have new DNA being synthesized. So 5' to 3'. So maybe what I'll do is I'll zoom in on this so I can make my drawing look just a little bit nicer on this. And I'm going to draw one more RNA primer over here because that is the way it's done. So 5' to 3'. So this one here, this Okazaki fragment was the first one. And this is the second one. This is the third one. And this is the fourth Okazaki fragment. So these are Okazaki fragments. Okazaki fragments. So making these Okazaki fragments, remember we have two enzymes. So we have, maybe I'll draw it as a red circle like this. We have RNA, the primase. So I'm just looking for a place to put the primase note. I'll shrink that here a little bit. Okay, primase. So primase has a short segment of complementary RNA. So where can we put that? We'll put it down here somewhere. So it says it adds a short segment of complementary RNA to the starting point. So it's a 3' OH end. And it looks like I made a typo there. 3' OH end for the DNA polymerase. So we've got our Okazaki fragments, and I'm almost done drawing this beast. Over here, we have another DNA polymerase. So there's my DNA polymerase. For the second fragment there, and almost all our notes. So let's take a look at that. We have one more thing to put on here, which is our primase. So we'll just check, find a new color for the primase. I guess I'll do that one in yellow. And the primase is over here. Shade that in just a little bit, maybe not completely, so you can see it. And this is the ligase. So the ligase joins those two Okazaki fragments together. So here we go. Ligase. Come on, there we go. Okay. Over here. And joins the two fragments together. So this one here, of course, is the lagging strand. So this is the kind of thing that, you know, when you're studying, I mean, I'm not going to ask you to describe all of the processes around DNA replication. But I'm definitely on the final exam going to be asking you something about DNA replication. So sometimes I might ask, what's going on with the leading strand? Tell me what all the enzymes do. I've also had a matching question that matches the enzymes with their functions. So haven't decided yet, but you can be guaranteed there's going to be some question about this. So I recommend when you're studying, draw this out. See if you can draw out what's going on with these different strands and figure out why all these enzymes are in there. I know that, you know, if you haven't seen this before, if you're not as familiar with it, the details are, there's a lot in there. And what I'm going to do next day is come back and we'll kind of review some of these enzymes and whatnot. So hopefully you got all that. I'll show you one more thing before I let you go today. So you can see there's my note to draw the replication fork on the board. And you can see that my diagram is, you know, maybe not as pretty as the textbook diagram. And I'll show you one more. Here's one that I've done on an actual whiteboard. You can see it's not much different. Maybe it's a little neater and easier to do on an actual whiteboard. But all the details are there. So like I said, make sure you go through and you need to know all of these enzymes and proteins. There's 6 of them. And like I said, there's going to be, that's at least 6 marks on your final exam. And probably a few more in terms of understanding what's going on with the DNA replication machinery. So it looks like we're out of time. Like I said, I'll come back and review some of these next day and show you a little bit of video, and maybe we'll do a Kahoot as well. So thanks for coming out, and I will see you on Wednesday.